Hello everyone. In this section, we will talk about where does genetic material exist. After watching this video, you should be able to tell, firstly, the location and form of genetic material, and secondly, types of plasmid. Generally, genetic material exists at 7 levels. They are cell, nucleus, chromosome, nucleotides, gene, genetic code, and nucleic acid. Another important existing form of genetic material is plasmid. A plasmid is a small DNA molecule within a cell that is physically separated from a chromosome DNA and can replicate independently. They are most commonly found as small circular, double-stranded DNA molecules in bacteria. Generally, plasmids can be classified into 5 types. The first one, conjunctive plasmid. Second one, resistance plasmid. The third one, bacterial-producing and antibiotic plasmid. The fourth one, plasmids with physical functions. The fifth one, toxin-producing plasmid. On the next, let's take a look at the properties of some classic plasmids. Please look at the picture on the screen. It is the genetic map of the E. coli F plasmid. The fertility factor allows genes to be transferred from one bacterium carrying the factor to another bacterium lacking the factor by conjugation. F plasmids are plasmids that can be inserted into chromosome DNA. Bacteria that have the F plasmid are known as F positive, and the bacteria without it are F negative. This picture depicts the genetic map of resistance plasmid, which is also called R factor. R factor is composed of 2 DNA fragments. One is called resistance transfer factor, or RTF. Another is called R determinant. Often, R factors code for more than one antibiotic resistance factor. The genes that encode resistance to unrelated antibiotics may be carried on a single R factor, sometimes up to 8 different resistances. Transfer of resistance factor among bacteria is one important reason that bacteria acquiring antibiotic resistance. The core plasmid, which contain genes that code for bacteriocins. Bacteriocins are proteins that can kill other bacteria and thus defend the host bacterium. Bacteriocins are found in many types of bacteria, including E. coli. Core plasmid can be grouped into 2 types. One group representative as core E1 is non-conjunctive and usually has low molecular weight. Another group representative as core IB is conjunctive and usually has high molecular weight. The Ti plasmid. It is called tumor-inducing plasmid. The Ti plasmid of agrobacterium tumescens could induce tumor-like growths on plants. The Ti plasmid serves as a natural vector in genetic engineering of plant cells because it can transfer its tDNA from the bacterium to the plant genome. How can we isolate plasmid from bacteria? Many methods have been developed to purify plasmid DNA from bacteria. These methods generally involve 3 steps. The first one, lysis of cell. The second one, removal of protein and RNA. And thirdly, purification of plasmid DNA. Identification of the plasmid can be conducted by electrophoresis. The DNA polymers of various sizes are separated by the molecular sieving action of the pores in the gel. DNA migrates differently depending on whether it is circular or linear. The linear DNA migrates through the gel pores in an end-on-end fashion and snakes its way through the pores. Circular supercoiled DNA is more compact and migrates through the pores more easily. The linear DNA almost always migrates more slowly than circular supercoiled DNA. Finally, let's take a look at the keywords in this section. Okay, that's all for this section.